Welcome back, and we were doing Viva questions, and we had done few questions, but I think few important and a simpler one are remaining, and those comes from genetic interactions. Like in the genetic interactions, you are given the experiment to study and work out the genetic ratio, and there I think you can very easily ask few very simple questions like who is father of genetics. I think you'll be able to answer that, who is father of genetics. Then next, which I thought you could be asked, is what are the Mendels Laws of inheritance? What are the 3 laws of inheritance which Mendel gave? Which is very easy to answer, but still it doesn't strike sometimes. Mendel gave 3 laws of inheritance, is law of segregation, law of dominance, and law of independence assortment. So Mendel gave 3 laws of inheritance, and the next question obviously comes is, what is law of segregation? What is law of segregation? Can anybody? So law of segregation is 2 alleles, and of the 2 alleles, only one allele is passed on to the next generation. That during meiosis they segregate. Only one allele is passed to the next generation. What is law of dominance? Is that law of dominance states that of the 2 alleles, one allele is dominant, and it is expressed, it is the only this allele which is expressed in the F1 generation. So he said in the hybrid progeny, one allele is dominant which is expressed in F1 generation. What is law of independent assortment? What is law of independent assortment? Is the alleles of 2 or more or different genes, they get sorted into gametes independent of each other. So the 2 genes, they're in the gametes, they assort independently. This is law of independent assortment. Though these may be similar, but it may not strike you, so you have to be prepared that these questions may also be asked in the exam. So can you quickly name the 7 characters which Mendel studied? Again, a very simple question, but you may not be able to answer, and you can get the answer from anywhere. I am not telling you here what are the 7 characters which Mendel studied. Then you may also be asked, what is a test cross? What is a test cross? The answer to this is when one heterozygous parent is crossed to the recessive homozygous, that is test cross, and you have studied this in earlier classes, so you don't need to worry for that. So then coming to the ratios, when do you get a ratio of 2:1? Like the common 5 ratios which you have in your syllabus, they would be easier to answer, but still sometimes to confuse you, you may be asked what is the ratio of 2:1. When do you get that? It is in case of recessive lethal alleles, that the one individual will die and you will get a ratio of 2:1. Recessive lethal alleles. Then few other questions which might be asked is what is epistasis? Then you need to answer. It's a very common question and it will be asked. What is epistasis? Epistasis is when one gene mutation affect the expression of other. When one gene mutation express the expression of the other, and it may be recessive mutation or it may be a dominant mutation. On the basis of that, the epistasis would be called as dominant epistasis or recessive epistasis. Then another very simple question which might come in between is that why was Mendel successful? And you know the reasons from your very early classes you are studying. But this a very simple question, but sometimes it doesn't strike, so you have to be prepared that it might also be asked. Then what is codominance? What is codominance? Is both the alleles, they are dominant. And any example for this is maybe the blood group. You can write that A and B allele, both of them, if they are present, they are codominant. Both are equally important in their expression. Then the next question which might be asked from my side is what is the difference between karyotype and ideogram? What is the difference between karyotype and ideogram? So karyotype is general, gives you the actual picture of total chromosomes in a cell, And in karyotyping, you study all the characters of chromosomes in a cell, all the chromosomes present in a cell and many characters. What are ideogram? An ideogram is basically a type of karyotype, and it shows the chromosome maps and with the bands, and they are arranged in a decreasing order. So it shows the chromosomes maps and they are arranged in decreasing order, and it is used in identifying the chromosomal abnormalities. But karyotyping is to study all the chromosomes present in a cell. And when do you get a ratio of 13:3? Meaning you may be asked this from any of the ratios. You have 5 ratios in your syllabus: 13:3, 12:3:1, 15:1, 9:3:4, and this 13:3. So when do you get a ratio of 13:3? 13:3 ratio we get in suppression epistasis, or where one gene is suppressing the other. The difference from dominant epistasis here is that the suppressing gene does not have a phenotype of its own, right? So it allows the other one. So because of this, we just have 2 categories: 13:3, where the gene is allowed to express, or gene is not allowed to express. So you get a ratio of 13:3 in suppression epistasis. So with this, I finish this Viva questions, and maybe I get some more questions if they come in my mind. I will come back with those questions any other time. Thank you.